Welcome back everyone to another ACE lecture. In today's lecture we are going to discuss the muscle system. So first I'm going to discuss different functions of the skeletal muscle. The main function of the skeletal muscles is to produce movements. And these movements are under our conscious control, but they also have the potential to function out of our conscious control. For instance, diaphragm, which helps us with breathing, is under our conscious control. We can speed up our breathing rate or slow down, but once we fall asleep, then the contractions and relaxations of diaphragm is out of our conscious control. In other words, this muscle undergoes automatic control. The other function of skeletal muscles is to help with maintaining body postures and to provide a stable stance during different activities, like for instance running or walking. Now let me ask you a question. What part of brain is required for coordination of movements as well as balance? So the coordination of muscle movements and balance are performed by cerebellum. Now moving on to other functions of skeletal muscles. Skeletal muscles can also be used to generate heat. So once our body temperature drops, skeletal muscles start to undergo contractions. In other words, they would cause shivering, as a consequence of which heat will be generated and will help to increase the body temperature. Skeletal muscles can also be used to mobilize venous blood. So in our circulation lecture, we discussed how the blood in the veins that are in lower extremities needs some help in order to be pushed back to the heart. And this system relies on different mechanisms. One, the venous system has one-way valves that prevents backflow of blood. There are also surrounding muscles, skeletal muscles, that will help push the blood upward towards the heart. And then finally, as we are breathing, there would be a negative pressure that is being created every time we try to breathe air in, and so that negative pressure again helps push the blood upward. So another function of skeletal muscles is to help mobilize venous blood, and it can also help to mobilize lymphatic fluids. So these were so far the functions of skeletal muscles. Now let's discuss the function of the cardiac muscle, which is used to pump blood into our systemic and pulmonary circulation. And then similar to skeletal muscles, these cardiac muscles are striated because they have a similar sarcomere structure, which we will discuss later in this video. And now regarding the smooth muscles. So smooth muscles are being found in the walls of hollow organs, like for instance it's being found in the pulmonary bronchi, and its function is to adjust the size of these structures. Like for instance, inside the pulmonary bronchi, it can constrict in response to allergens to prevent further allergens getting inside our lungs. Or another example is smooth muscles inside our blood vessels that can constrict and increase blood pressure. So examples of smooth muscles are in bronchi, is in our blood vessels that can constrict and elevate blood pressure. Another example is in our digestive system, for instance, that can contract and move food via peristalsis. And then another example is smooth muscles in the bladder that help with urination. Okay, so next let's move on to the types and characteristics of these muscles. So we've already discussed that both skeletal and cardiac muscles are striated. In addition, skeletal muscles have multi-nuclei. So here you can see that one skeletal muscle can have multiple nuclei versus cardiac muscles that can have one or two nuclei. And then smooth muscles have only one nuclei. Skeletal muscles are non-branched, they're straight, versus cardiac muscles that have branches. And then smooth muscles have a spindle shape. So here you can see that they have spindle shapes. There are also gap junctions that is present both in smooth muscles and cardiac muscles, and these gap junctions allow the electric potential to spread rapidly from one muscle to another. And this way there would be a rapid coordinated contraction of smooth muscles as well as cardiac muscles. Skeletal muscles, however, do not have gap junctions. Regarding contraction, we discussed that skeletal muscles are under our voluntary control versus cardiac and smooth muscles are not under our conscious control. In terms of location, skeletal muscles can be either attached to bones like our triceps or biceps muscles, or it could be attached to the skin like, for instance, our facial muscles. Cardiac muscles, as the name implies, construct the walls of the heart, and then smooth muscles we discussed, they're found in the walls of hollow or visceral organs like, for instance, bladder, GI tract, and blood vessels. Regarding the mode of contraction, we will discuss how calcium affects troponin in both skeletal and cardiac muscles in order to induce contraction, versus there would be activation of calmodulin in the cytosol for the smooth muscles. Are there pacemakers that are present? Yes, for both smooth muscles and cardiac muscles there are pacemakers. And pacemakers are basically referred to cells that will initiate an action potential. So if you cut all the parasympathetic nervous system and sympathetic nervous system connections to the heart, these heart cells can still continue to pump blood because they will initiate their own action potential, and so that's referred to as presence of pacemakers. So is with the GI tract. GI tract still will undergo peristalsis even if you cut the connections. So then you may ask, then what's the role of the parasympathetic nervous system or sympathetic nervous system? Well, these autonomic nervous systems will regulate the rate of these pacemakers. For instance, in the heart, sympathetic nervous system will signal the pacemaker cells to increase their firing rate, versus the parasympathetic nervous system lowers the firing rate. So therefore, that's why the parasympathetic nervous system decreases heart rate while sympathetic nervous system increases heart rate. Same happens with the smooth muscles. SNS will activate them, as a consequence of which there would be constriction of the blood vessels, and so therefore there would be elevated blood pressure, versus the parasympathetic nervous system that, for instance, acts on the urinary bladder and induces its contraction in order to promote urination. So we do have a lecture on the nervous system, inside which we will discuss how the sympathetic nervous system and parasympathetic nervous system will affect different systems. So we will compare them in a table showing you how different organs are affected differently by every one of these systems. Now, regarding the nervous control, just discussed that autonomic nervous system, which is composed of SNS and PNS, can regulate both the smooth muscles and cardiac muscles, versus the skeletal muscle that is under the control of the somatic nervous system that induces activation of motor neurons. Now, regarding speed of contraction, skeletal muscles can vary, and it depends on what type of muscle we are talking about, fast twitch versus slow twitch. So the speed of contraction can vary for skeletal muscles. Cardiac muscles have a relatively slow contraction speed, and then smooth muscles have a very slow rate. So think of contraction of the GI tract, like for instance. It takes much longer for them to contract in comparison to the heart that is contracting. Okay, and then regarding their mode of respiration, whether it's aerobic respiration or anaerobic respiration, skeletal muscles rely both on aerobic and anaerobic respiration. Cardiac muscle relies on aerobic respiration, and then smooth muscles rely mostly on aerobic respiration. Now next, let's discuss the structure and the mechanism of contraction of the skeletal muscles, and then following that we will discuss the sarcomere structure. So in order for a skeletal muscle to contract, it must first be stimulated by a motor neuron. Alright, so we already discussed motor neuron is under the control of the somatic nervous system. And the cell body of these motor neurons are located in the central nervous system, like for instance brain and spinal cord. and their axons run all the way to the periphery. The ending of these axons are called a synaptic bulb. So here you can see that we have a synaptic bulb, which is the ending of the axon for a motor neuron. And these synaptic bulbs lie very close to the surface of the muscle fiber, and this surface is known as motor end plate. And then the space between the synaptic bulb and the motor end plate, so here we have the synaptic bulb and then the motor end plate here. The space in between is called a synaptic cleft, which is basically used to relay information from the nerve to the muscle with the help of the neurotransmitters that are being released. And in this example, by motor neurons, it's acetylcholine that is being released. So what happens after acetylcholine binds to the postsynaptic receptors is that it will cause opening of the sodium channels, as a consequence of which now there would be a depolarization wave that will be initiated on the surface of the muscle fibers. Now these depolarization waves can go deep into the muscle fiber by special structures that are called T-tubules, which are basically invaginations in the cell membrane. So here we have T-tubule, which is invagination in the plasma membrane. And consequently, the action potential in the T-tubules will act on dihydropyridine receptors to further act on the ryanodine receptors to induce release of calcium from sarcoplasmic reticulum. Alright, so here we have dihydropyridine receptors, dihydropyridine receptor that will interact with the ryanodine receptor, and from here on there would be release of calcium from the sarcoplasmic reticulum. Alright, so just to quickly summarize, there was an activation of a motor neuron, as a consequence of which acetylcholine was released in the postsynaptic cleft. The acetylcholine then binds to the postsynaptic receptors and cause opening of the ligand-gated sodium channels. Consequently, entry of sodium into the cell causes an action potential. And this action potential on the surface of a muscle fiber can go further deeper into the inside the muscle with the help of the T-tubules, which are special structures that are being created from invagination of the plasma membrane. Now this depolarization wave acts on the dihydropyridine receptor as well as the ryanodine receptors in order to induce release of calcium from sarcoplasmic reticulum. And then following release of calcium, calcium binds to troponin in order to act on tropomyosin to move away from the actin binding site, as a consequence of which now myosin can bind to actin and induce contraction of muscle. And I'm going to discuss this in a little more detail with you and show you images, but before I go that far, first I would like to show you the structure of myosin and actin within the muscle fibers. So here I have drawn you a structure of a sarcomere, which is the basic unit of skeletal muscles that runs from one Z line to another. So here we have one sarcomere. And it's upon contractions that these Z lines will become closer to each other, as a consequence of which the whole length of the muscle will decrease. So how does this happen? By having the actin and myosin sliding on top of each other. And I will show you the mechanism in a little bit, but first I would like to discuss the structure of the sarcomere in a little more detail, because you're expected to know these names. So basically the Z line is sarcomere end-to-end junction where the thin actin filaments join. So here the thin lines, these are actin filaments. and Z line is the ending of a sarcomers where the actin filaments join. Then we have the M line, which is made of a thick myosin fibers that join at the center of the sarcomere. The whole length of myosin will generate a dark color, and this whole length is referred to as A band. H band, on the other hand, is a fairly lighter area because there is no overlap between actin and myosin. Okay, and then we have the I band, which is only made of the actin filaments, which is the light bands, and it has a lighter color. So you can see how there are color differences, dark versus light, that keeps on going in alternative orders. So that's why there is a striation color that is present in both skeletal and cardiac muscle. All right, so with that background in mind, let's continue from where we left off. We just discussed how calcium was released. So now let's discuss how calcium affects actin filaments in here. So here you can see that we have a myosin fiber and an actin fiber that is made of two fibers that are twisted together. Now on the actin we have two proteins. One is called tropomyosin, which covers the binding site of myosin on actin. So it covers binding site of myosin to actin. So therefore, under relaxed state, like the example shown right here, the myosin fiber cannot bind to actin because tropomyosin is covering that binding site. So that binding site is again present on the actin that is being covered by tropomyosin. As a consequence of which, myosin cannot bind to actin. Now what happens when calcium is released is that it binds to troponin, which is another protein, and now troponin can act on tropomyosin to change its conformation and move away from myosin binding site. So now tropomyosin has moved away from the binding site, as a consequence of which now myosin can bind to actin. So this is the very first step that is required for calcium, to allow myosin to bind to actin. After this binding, this head of myosin is charged with a lot of energy. It can undergo a phenomenon called power stroke, where it bends and it helps slide the actin filament. And so by this power stroke, it allows the actin filament to slide on top of myosin and cause contraction of the muscle. All right, and then in the next step, myosin will not just release from actin. In order for myosin to let go of actin, ATP molecule needs to bind to myosin. And then from there on, the energy within ATP is used to recharge the head of the myosin in preparation for the next coming up contraction. So let's one more time explain what had happened here. So under relaxed state, there is a binding site for myosin on actin, which is blocked by tropomyosin. Once calcium is released, calcium will bind to troponin to activate troponin. And now troponin acts on tropomyosin and change its conformation, as a consequence of which now the binding site on actin becomes available. So now myosin can bind to actin. There is energy that has been stored in the myosin head, and this energy will help to initiate a power stroke, which allows the actin filament to slide on top of myosin and cause contraction of the muscle. Next, in order to release myosin from actin, ATP molecule has to bind to myosin, and then the energy from ATP molecule is used to charge or energize the myosin head for the next upcoming contraction again. So now let me ask you a question. Why is it that people who die within a few hours, their body become very stiff? So this phenomenon is actually called rigor mortis. And the reason for that is that after someone dies, within a few hours, the ATP supply will be all depleted, as a consequence of which the myosin cannot release actin, and that's why that this cross bridge will cause stiffening of muscles after death. So as you can see, ATP has a very important role in the muscle system. So in addition to producing energy to allow actin and myosin filaments to slide on top of each other, ATP also has other roles. And these include, for instance, it will help with providing the energy for the Na plus K plus ATPase pump, which basically pumps, if we have a cell here, it pumps 3 Na plus outside the cell in exchange for 2 K plus that comes inside, and this is being done with the help of the Na plus K plus ATPase pump, which uses ATP. So what happens now is that outside environment has more positive charges than inside, as a consequence of which the cell membrane will have a negative potential. And that's actually the basis of initiating a depolarization wave, because normally outside is more plus and inside is more negative. Once Na plus start to enter, then that's the time that we have more plus inside, and that's where the depolarization wave will go through. Okay, so that's another function of ATP. And then ATP is of course used to pump calcium into sarcoplasmic reticulum. Okay, so here how I've shown you that we have all these calcium that is being stored in the sarcoplasmic reticulum. It's pumping inside the cell, is being done with the help of the ATP. And then regarding the sources of ATP, in the first few seconds of exercise by a muscle, we have ATP sources that will quickly deplete, and then the muscle will rely on creatine phosphate in order to convert ADP into ATP molecule. After a few seconds for energy production, now the muscles have to rely on anaerobic glycolysis. And if the muscle wants to last much longer, then that's the time that it has to rely on aerobic respiration as well as fatty acid oxidation for providing energy supplies. And in fact, these modes of energy production provides the basis for the difference in the contractile force of different muscles. And so that's actually the next topic that we would like to discuss, classification of different skeletal muscles. So once you go to restaurants, a commonly asked question is that, do you prefer red meat versus white meat? And the reason that the red meat has a red color, which is shown right here, is that this type of meat has many blood vessels and has a high myoglobin content. So basically myoglobin is a red protein that contains heme molecule that helps with the storage of oxygen in the muscle cells. So you know how hemoglobin helps with storage and carrying the oxygen inside the red blood cells? Here we are talking about myoglobin, protein inside muscles that helps with the storage of oxygen. So because of high myoglobin content and many blood vessels, the type 1 or slow twitch muscles have a red color. All right, and these muscles have a lot of mitochondria as well as myoglobin content to have high oxygen content in order to initiate aerobic respiration. So in other words, slow twitch muscle fibers are mainly using aerobic respiration versus fast twitch that uses anaerobic glycolysis for energy production. Now this difference in the source of energy production will generate different properties for every one of these muscles. So here we have red fibers versus here we have white meat or white fibers. And so as we just discussed, red fibers rely on aerobic respiration. They can use triglycerides for energy production, as we just shown in the previous graph. Consequently, these muscles are very resistant to fatigue and are highly efficient. They can last for many hours. So you see how human runners, since they are having red muscle fibers, they can run for hours. But then the downside to these muscle fibers is that their contraction time is relatively slow and the maximal force that they produce is moderate. Versus the fast twitch muscle fibers, or also known as type 2 muscle fibers, which rely on anaerobic respiration. Since they don't have many blood vessels and they don't rely on too much mitochondria and myoglobin, therefore they have a white color. They use creatine phosphate and glycogen for energy production. So they rely mainly on creatine phosphate and glycogen in order to supply the glucose for anaerobic glycolysis. And in return, they have a very high contraction force as well as a fast contraction time. However, these muscles are, in comparison to the red fibers, they can become fatigued very quickly. Indeed, we have two types of white fibers, type 2A, which can last for less than 30 minutes, and type 2B, which is kind of like the extreme form of fast twitch muscle fibers that will last less than 1 minute. And then, as we just discussed, these muscle fibers have a low mitochondria and myoglobin content because they primarily rely on anaerobic respiration. Now, a concept that I would like to discuss here with you is called oxygen debt. So, oxygen debt. So as muscle will exercise, their oxygen supply will get lower and lower, and so therefore the muscle will become fatigued, meaning that it will get slower and weaker as we make it exercise more and more. And this is due to the fact that as the oxygen supply of these red muscle fibers decrease, therefore their energy production will become more inefficient. And so therefore now they have to rely on anaerobic respiration, as a consequence of which there would be buildup of lactic acid in our muscles, which gives us that burning sensation when we overuse our muscles. And so that's the reason that red fibers have to have a lot of myoglobin and mitochondria in order to maintain an efficient contraction mechanisms. Otherwise, if the oxygen supply drops low, then that's the time that there would be inefficient energy production and lactic acid buildup. All right, the next concept that we would like to discuss is cardiac muscles. And similar to skeletal muscles, cardiac muscles also contain sarcomers, as a consequence of which they have striated appearance. And then another difference we just discussed, cardiac muscles contain their own pacemakers, which allow their autorhythmic contractions that can be regulated by the autonomic nervous system. All right, so autonomic nervous system, like for instance, SNS increases heart rate, while PNS, like for instance, vagus nerve, decreases heart rate. Another important difference between cardiac muscles and skeletal muscles is the presence of gap junctions in cardiac muscles. So there are structures called intercalated discs where the plasma membrane of two adjacent cardiac cells are held together. And mechanically, these two cells are held together by desmosomes, while their cytoplasm is connected to each other by gap junctions. As a consequence of which, gap junctions will now allow an impulse to get easily transferred from one cell to another one. So consequently, considering that cardiac muscles are branched and they have gap junctions, therefore there will be a uniform and continuous contraction of cardiac muscles. The next muscle I would like to talk about are the smooth muscles. And basically, smooth muscles are found in the walls of hollow organs, as we just discussed, like for instance, urinary bladder, blood vessels, GI tract, respiratory tract, and so on. But then in comparison to the skeletal muscles, smooth muscle relies on a very small amount of energy and contracts at a much, much slower rate. Like for instance, look at the peristalsis of a GI tract, how slowly it goes through, right? So the rate of contraction of smooth muscles is very slow and needs little energy. And so consequently, since very little energy is needed and there won't be any explosive amount of exercise by these muscles, therefore mostly aerobic respiration will be used by smooth muscles to generate energy because there is usually no oxygen debt in these muscles. Another important feature of smooth muscles, since they do little work and they need little energy, these muscles generally, unlike the skeletal muscles that can fatigue, these muscles do not generally fatigue. Okay, of course there are examples that if the blood supply is short in some of the GI tract, then that's the time that it would cause fatigue of those muscles and the patient will feel pain in that area. But generally, if there is a normal blood supply, then these muscles will not fatigue because they have a very slow contraction and do not do a lot of mechanical work. Now, in terms of their structures, similar to the skeletal and cardiac muscles, smooth muscles also have both actin and myosin filaments. And then these filaments attach to a structure called dense bodies, which is analogous to Z discs in the cardiac and skeletal muscles. So here you can see that we have the dense bodies, and then we have the actin filaments, and then myosin on top of that. And similar again to the cardiac and skeletal muscles, there would be sliding of actin and myosin on top of each other in order to induce contraction of smooth muscles. Now, the mechanism of contraction of smooth muscles is different than skeletal and cardiac muscles, and the two main differences is that one, troponin is not present in smooth muscles, and therefore the binding site of myosin to actin is always open. And second, the initial calcium supply is from extracellular space. You know how we discussed that for skeletal and cardiac muscles, it's endoplasmic reticulum that is releasing calcium. For smooth muscles, the initial supply of calcium is from extracellular space. So extracellular calcium is being used. So now let's discuss the mechanism of smooth muscle contraction. So following depolarization of the smooth muscle, there would be activation of the calcium channels that will allow influx of extracellular calcium into the cell. And then there will be later on further release of calcium from endoplasmic reticulum. Once calcium concentration increases in the cytoplasm of smooth muscles, calcium binds to calmodulin to form a complex that will now activate the myosin light chain kinase. And the job of the kinase enzyme is to phosphorylate myosin fibers. So therefore, now a myosin fiber will become phosphorylated, and this phosphorylated chain can cross-link to the actin filament. All right. So since there is no troponin, we don't have to wait for the tropomyosin to change conformation. Once the myosin light chain is activated, it's now able to cross bridge and bind to the actin filament, following which there would be smooth muscle contraction. And then, as we just discussed earlier, similar to cardiac muscles, smooth muscles also have gap junctions to allow coordinated and uniform contraction of the smooth muscles. All right, so that concludes our discussion of the skeletal muscles, cardiac muscles, and smooth muscles. We've already discussed mechanism of contraction for different one of them. We discussed the types and characteristics of different skeletal muscles, including their energy sources, and we also discussed functions of cardiac, skeletal, and smooth muscles. So next, what I would like to do is answer some of the questions that I have not answered in this lecture, and then following that, we will go over some practice questions. So the very first question is, why do skeletal muscles contain multiple nuclei? And the answer is that during embryonic development, myoblasts, so anytime you hear the term blast, it means the immature form of that type of cell. So myoblasts, multiple one of them fuse to each other in order to form a muscle fiber. And so therefore, since multiple myoblasts are combined together, therefore there would be multiple nuclei. Okay. The next question is, are skeletal muscles able to regenerate themselves? Generally, the answer is no. So basically, when you exercise and you notice that your muscle keeps on getting bigger and bigger, it's not because they increase in number, but rather it's because they increase in size. But then under extreme conditions where there is extensive damage to the skeletal muscles, there are some stem cells that are still around, and these stem cells are called satellite cells, which can generate skeletal muscles. All right, but under normal conditions, you don't usually see the skeletal muscles regenerating themselves. What is the definition of a motor unit? So basically, a motor neuron plus the muscle fibers that are under the control of that motor neuron, these are called a motor unit. So here, the example that we went through, this was an example of a motor unit. Okay? Next question. What is the difference between sarcomere, myofibril, and muscle fibers? So we've already discussed sarcomere. We mentioned that this is the basic unit of striated muscles. Myofibrils, on the other hand, these are basically referred to the arrangement of sarcomeres that are in series. So if you put multiple sarcomeres in series, that is called a myofibril. And then muscle fibers is arrangement of myofibrils in parallel. So once you put multiple myofibrils in parallel, that's the time that you will form a muscle fiber. Describe the sliding filament model. So basically this is what we've been explaining regarding the sliding of actin over myosin. So based on this model, muscle will shorten their length during contraction by sliding of myosin and actin filaments on top of each other. The next question is, after depolarization, how does tropomyosin resume its initial position to prevent binding of myosin head to actin? So what happens after depolarization? After depolarization, calcium will start to get pumped back into sarcoplasmic reticulum, as a consequence of which the cytoplasmic concentration of calcium drops, and so therefore troponin will no longer act on tropomyosin. And so tropomyosin will resume its initial position to block binding site of myosin on the actin filament. Now let's go over some practice questions. I'm going to read these questions for you, and then you can pause the video to answer the questions, and when you're ready, then you can resume the video. So which of the following muscles are under our conscious control? The next question is, increased activity of which of the following muscles directly causes vasoconstriction? Which of the following neurotransmitters are used to induce muscle contraction via motor neurons? And then finally, which of the following is the feature of type 1 muscle fibers? All right, so answering these questions should be easy after this lecture. Which of the following muscles are under our conscious control? The skeletal muscles, these two are not. Okay, so choice A would be the correct answer. Next is, increased activity of which of the following muscle directly causes vasoconstriction? So inside the blood vessels, what type of muscles do we have? And the answer is smooth muscles. So smooth muscles are responsible for directly inducing vasoconstriction upon action of the sympathetic nervous system. Which of the following neurotransmitters are used to induce muscle contraction via motor neurons? And actually, here I have the answer for you. We just discussed how acetylcholine is used by motor neurons. So the answer is acetylcholine. And let me ask you one other question. What other autonomic nerves rely on acetylcholine as their neurotransmitters? So you know how we have SNS and PNS, then we have ganglionic nerve and then post-ganglionic nerves. So both the pre-ganglionic nerves of PNS and SNS rely on acetylcholine. But then the post-ganglionic nerves are the ones that are actually inducing effect. So with the PNS, acetylcholine is the main neurotransmitter of PNS, versus with the SNS or sympathetic nervous system. Can you tell me which neurotransmitter is the main neurotransmitter for SNS? Norepinephrine. Okay, so SNS induces fight or flight response versus PNS induces rest or digest response. And then the final question is, which of the following is the feature of type 1 muscle fibers or red muscle fibers? They are highly fatigue resistance. That's correct. They have high mitochondria content. That's correct. They also have high myoglobin content because they rely on aerobic respiration. And then fast contraction time, that would be incorrect. Okay, because we just discussed in comparison to white muscles of, for instance, like cheetah, that is much faster than our red muscles. The type 1 muscles have a slow contraction time. So type 1 is red fibers have a slow in comparison to type 2 muscle fibers, which are white fibers, like in cheetah, which is very fast, but they get fatigued really quickly. They don't last long. So therefore, choice C would be the correct answer. All right, and that concludes our discussion of the muscle system. Hope you found it helpful, and we will see you for the next lecture.